Welcome to this Antibody 101 video series. Our goal is to highlight the high-level concepts for antibodies in particular and proteins in general. My name is Kathy. I have a PhD in synthetic biology from Stanford. I trained at the Institute of Protein Design as a postdoc, and I've worked as a scientist in biopharma for many years now. In this video, we'll cover Antibody 101, Part 3, which is to answer the question, what exactly holds an antibody or a protein in general together? All right, first we're going to start with the quaternary structure of a protein. And this is kind of the representation I've been showing in the antibodies, where there are multiple chains, remember there are 4 in an antibody, that come together in a particular shape, sort of by magic. Now, if you zoom in to a single chain, then we're talking about the tertiary structure. So if you took the 4 chains of an antibody and pulled them apart, you'll have 4 separate units that all fold, again, pretty magically, into a particular shape. Now, if you zoom in even further, as sort of illustrated by these squiggly black lines in this figure, there are particular patterns of the backbones of the protein chains. So in particular, there are 2 types that are very famous and very common. First is the pleated sheet, which is also called the beta sheet, and sometimes just called a sheet. And then the second is an alpha helix, also just called a helix. And these are considered the secondary structure elements. Now, if you zoom in even further, now we get to the primary sequence of amino acids that make up a protein. Now, if you'll recall, a protein, an antibody is a protein, is made out of basically a string of amino acids on a bead. And they are one after another. And it's the properties of these amino acids and how they come together that then form the secondary structure, that then form the tertiary structure, that then form the quaternary structure. All right, here are all of the common amino acids, and I will leave this here mostly as a reference so that you can pause the video and go back and look at these. But essentially, we'll look at the properties of these grouped into 3 different types of interactions. So the first interactions are essentially kind of like glue, right? They're going to kind of hold things together. It's a little bit sticky, but it's a little bit non-specific as well. The second type of interactions are ionic interactions, and you can think of them as magnets with varying degrees of strength. So here you get kind of action at a distance, and just like with large magnets, positive is attracted to negative, and positives will repel, and negatives will repel. And then the last type are staples, which are formed in a particular way and actually staple 2 parts of a protein together. Okay, in terms of glues, at the molecular level, this is hydrophobic interactions. And this is one way to categorize the amino acids. So there are, as you can see, many amino acids that fall into this category where if they come close to each other, they can kind of form a sort of gooey interaction where they're a little bit sticky, but it may not be the strongest or most specific force. So here are the amino acids that are charged or partially charged, and they can interact in these kind of ionic ways, then behave like little miniature magnets. And in the last category, cysteine is a special amino acid. When 2 cysteines come together at a specific geometric orientation, they can form a stable bond that essentially staples that part of the protein together. Okay, so let's go back to the carbon-carbon sort of glue-like hydrophobic interaction. Now, if you look at a unit of a protein, in particular, this is the VH domain of an antibody. So we've zoomed in on the claw of an antibody, and then we've zoomed in on the tip of that claw, and then we're only taking 1/2 of that tip of the claw. So that is the VH domain that you're looking at here. So when we represent it in this cartoon diagram, you can see that there are these rectangular blocks, and those represent the sheet secondary structure. But when it's drawn this way, it basically looks very empty. So it's kind of mysterious looking, like why this can hold together in this particular shape. But in actuality, if we go to a different representation, specifically a sphere representation of all of the atoms that are involved here, you can see that a very different picture in that there is very, very actually little space in the middle of this protein. So on the right diagram here, the white spheres come from kind of this sheet in the back, and the black spheres come from the sheet in the front. So in this particular domain, you can think of two sets of sheets, kind of like pieces of bread in a sandwich. And in the middle are all of these atoms that are a little greasy. So you can think of it as butter. So it's butter that's holding together two slices of bread. And this is actually a real fold in proteins. It's called a sandwich fold for this reason. Okay, so the next category of interactions are these little magnets, right? So at the molecular level, we have nitrogens and oxygens that can interact and form basically a little magnet in your protein. And here I'm just demonstrating one of these such interactions in the sphere representation, where you can see, they are very, very much right next to each other, pretty much touching. And that's how you get these little magnetic interactions within your protein. And unlike the glue interactions, which are pretty non-specific, so you can get slippage, these ionic interactions are a little bit more specific because there's an orientation to them. And so on the right, I'm showing just the backbone atoms in more of a stick representation so that I can see this better. And the yellow lines highlight all the places where there's basically a little tiny magnetic interaction between everything in these backbone atoms. And this is actually one of the reasons that this is called a sheet, because it forms this very particular ladder pattern of these little magnetic interactions. And that's not true just for the sheets. The sheets form a particular pattern in the backbone of these ionic bonds, but helices as well, their backbones, when they spiral around like this, also form a very, very distinct pattern. And this is why these two secondary structures are so common. Okay, and then the last type of interaction that we're talking about here are these staples. So these only happen between two cysteines, and they can only happen if they are exactly the correct distance and orientation from each other. But when they are like that, it actually forms a covalent bond. So this is why I call it a staple. It literally takes those two parts of the protein and physically sticks them together. So this is a very strong interaction that holds this part of the protein on the left to this part of the protein on the right, only in this very particular orientation. Okay, now backing out, all of the interactions I've shown are within a single protein chain, so that's the tertiary structure. But the exact same forces work on the quaternary structure, which is between different protein chains. Recall that the antibody has 4 different chains, and so here we're highlighting kind of the locations of where all the glue-like interactions between different chains occur, and then where all of these little magnetic interactions occur between the chains, highlighted in yellow in the middle. And then on the right, the locations of staples that are specifically between the different chains of an antibody. And so this is not a completely quantitatively realistic representation of the interactions, but you'll get a sense that typically these interactions between different chains are not as extensive as the ones that are within each chain. And that reflects physical reality, right? So you can pull different chains of a multi-chain protein apart, and they will still form their individual chains. And that reflects kind of the amount of interaction between those two types of interactions. Okay, with that, thank you for learning with us today. Biology breaks every rule that it makes, so remember that what is presented here is one way that things work, and there are many different ways that could work. Please like and comment, and see you in the next video.